Now the time has come for the Michael Davis Memorial Lecture, which will close this excellent day of scientific sessions. A little bit of the icing on the cake. For those of you who have not been lucky enough to have met Michael Davis, Mike was the professor of cardiac pathology at St. George's. He was an excellent scientist, really and truly a wise man. His contribution to the understanding of the acute coronary syndrome was really, really superb. We changed the way we thought about mechanisms of the acute coronary syndrome after his sort of pathological investigations. We could therefore not think of a better person to invite to give the Michael Davis Memorial Lecture today than Professor Keith Fox. Keith is one of the most influential cardiologists of his generation. We have a summary of his millions of achievements and over the years in our program, so you can actually enjoy reading those incredible achievements. I will not have sort of time to actually go through the whole list. It's only just to say that you find very few people in whom scientific knowledge and clinical knowledge actually amalgamates in such a balanced way. And this is really something which I have valued over the years. His contribution to understanding acute coronary syndrome has been really very important. And we are seeing the disease in a different light after reading his work, and also from a management point of view, again, that his work has influenced the way acute coronary syndrome is actually treated around the world. He has come back from India only a few hours ago. He must be destroyed. So I'm extremely grateful for you, Keith, to actually be here today with us. And it's a great pleasure and a privilege for me to invite you to come to the lectern to deliver the Michael Davis lecture.
Thank you very much.
Pleasure.
Juan Carlos, thank you so much for the invitation. I'm delighted and honoured, really, to be able to give this lecture. And you know, you've heard a lot today about some of the tremendous innovation that there's been in the field of thrombosis and antithrombotics. And there are people out there who say, well, you know, the management of acute coronary disease has now reached a peak. It's all going to be downhill from here because we've done all that we can do. And I want to put to you a new paradigm that we haven't. And I'm going to take a slightly different view. And before doing that, I want to pay tribute, just as Juan Carlos has, to Michael Davis. All of us who knew Michael Davis are aware not only of his key observations and his insights, but his generosity of spirit in how he was prepared to share these with all of us and make these freely available. And that's a message about how science should be. And importantly, he set out unresolved questions. So here are my disclosures, both in terms of various obligations and the funding. And most of our funding is from those listed here. So for those of us in the room, is it all too long? Is it too late? Because atherogenesis starts early. This is a 32-year-old female who was a heart transplant donor. And you can see that her plaque is considerable at the age of 32, and she was not either homozygous or heterozygous FH. In fact, if you look at donor age and the prevalence of atherosclerosis, this is very worrying, because you can see that by the time you haven't reached 30 yet, 37% of these individuals have already got atherogenesis. So if we're to implement studies or implement treatments that are going to change the pattern of atherogenesis, we have to do that very early if we're going to do it. Otherwise, to think about other targets. And my friend and colleague Salam Yousuf says it's all explained by risk factors. And I helped design InterHeart. And in InterHeart, the analysis was that 2/3 of the population attributed risk is accounted for by smoking and abnormal lipids. And that 90% is accounted for by about 9 modifiable risk factors. And these are listed below. However, I disagree with that interpretation. And let me explain why. This shows you the analysis from InterHeart and the different risk factors and how they all accumulate. And statistically, they do. The problem is that the observational data may overestimate the treatment effect. Next, the risks may not be additive. Next, the controls have substantial absolute risk of coronary heart disease, and there could be causal mechanisms as well as bystander effects. One of the other presenters at the Cardiac Society of India was Bob Roberts, who is leading some of the genomic studies, and he says half of the coronary artery disease is because of genomic factors. Are these two irreconcilable? The answer is no, because of the fact that there are ways in which environmental factors via inducible forms of RNA may modify the expression. Let me show you this, just as an aside, a cartoon. It's a nice teaching cartoon. It shows an artery, the development of a plaque, a lipid-rich component, protrusion, rupture, thrombosis, and bad news. The trouble is, it's wrong for several reasons. It's wrong because of the fact that it shows this as a continuous process when actually it's not. It's wrong because it shows no adaptive remodeling. And this picture, which comes from one of our late patients, is a far closer representation of the paradigm. And this shows complex atheromatous disease of the kind that Michael Davis clearly defined, that this individual had a lipid-rich plaque that ruptured, and at that time, the angiogram showed that the individual did not have obstructive coronary disease. So there was no lesion to revascularize. There was no obstructive lesion. Even an FFR would not have defined that. And unfortunately, they did not adhere to some of the secondary prevention measures, and the plaque grew, re-ruptured, and the patient died a sudden death. So what we have is not a continuous gradual process, but a stochastic process that has abrupt changes as the disease progresses. Next paradigm. Let's look at this EM micrograph of a coronary artery. What you can see is some normal cells out here, endothelial cells. But you can see disruption of the endothelium with exposure of the subendothelial matrix. You can see mononuclear cells that are invading and adherence of some platelets. And I want to put to you a different paradigm. This paradigm is that in our textbooks, plaque rupture was a rare event and pretty devastating. And I'd like to put to you that erosion and rupture are not rare, they're actually common, but mostly they heal. And let me cite some of Michael Davis's data to support that. I'm putting to you this concept that plaque rupture is neither rare nor singular, and there's a balance between progression and repair, and this is potentially modifiable. And both Mike Davis, Falk, and others demonstrated that in people who were incidental victims, in some cases road traffic accidents. In some cases, they were victims of war. But these were people who were the passengers. They were not the drivers. And incidentally, about 9% of them had an erosion or rupture in a coronary artery at the time of their death, despite the fact that this was incidental and amplified in diabetics and hypertensives. So that tells us that this is not actually very rare. And those of us in the room, males over 45 and women over a little bit older age, have probably got these erosions in our coronary arteries at the moment, at least in a proportion of us. And we just hope the repair mechanism is in balance and that your pudding doesn't disrupt it in the wrong direction. So Mike Davis based a lot of his histology on very astute observations. So here's an observation of a diseased coronary artery, and you can see two complex plaques. You can see a plaque here that's ruptured, a lipid-rich plaque, thin fibrous cap, thin fibrous cap. However, are these the same? These are in the same plane, in the same individual. Which of these, or both, is susceptible to re-rupture? Well, if you look at the inflammation in the plaque, it's entirely different. There's CD68 in one of these, but not the other. So in the same individual, in the same plane, we can have two plaques, one of them with upregulated inflammation that is inducing caspases that's breaking down the structure of the plaque, and the other not. So the idea that we can predict just based on structure alone, I put to you, is outdated. What's the next evidence? It comes from several studies that have shown that there are multiple sites of plaque disruption and inflammation, and that this is a repetitive and usually silent process. And this I'm just showing you from some of the Japanese data. Now, if we were to look at the angiogram, we would look at the angiograms, which are not very well shown in this illustration, and pick up what is thought to be the culprit lesions. However, elsewhere in the coronary artery, we couldn't do angioscopy through our ethics committee in this country and many countries elsewhere in Europe. But what this shows is that there were several areas where there were angioscopic lesions unrecognized. So multiple lesions in the same individual at the time of the culprit lesion, which is number 8. Now, we've demonstrated in the GRACE program that if you survive to get into hospital alive, not surprisingly, those with ST elevation have got biggest risk. But what is perhaps surprising is that if you survive the first 15 days, those who presented as non-STEMIs actually have the greatest long-term risk. And this is even after adjusting for the differences in age and baseline characteristics. And the non-STEMIs presenting with ST depression versus these presenting with ST elevation, and these with ACS with documented coronary disease but no ST deviation. So what does it mean? Well, with a collaboration between the UK and Belgium, we undertook this study looking at long-term outcomes. We've got some unique advantages in several of our European countries by being able to link long-term data with the acute information with no missing individuals. And what this tells us is something quite interesting. As cardiologists, we focused on the acute phase, but there's something else going on. That if your patient turns to you after they're non-STEMI and says, okay, I've survived that, I'm going to be all right now, doc, aren't I? And what you should tell them is, of their 5-year risk, 85% of it is in front of them still, despite the current standards of care in our European countries. And they were highly applied in secondary prevention. 86% of the deaths were after discharge. So I think we focused not enough on the longer term phase and too much, well, we've achieved a lot in the acute phase, but not enough in the long-term phase. Secondly, in trials, we rightly focus on the time to the first event, because that is the least confounded. But what our patients are concerned about is the time to all their events, not just the first event. And here I show you, in our complete 5-year cohort, the individuals who had not just one myocardial infarction, but multiple, several revascularizations, several strokes, and several unstable angina admissions with an abnormal ECG. So people have repeated events, repeated hospitalizations, and there are multiple lesions. And this is supported by data from the study led by Greg Stone, the PROSPECT study, where intravascular ultrasound was conducted to look at culprit lesions and other lesions. And what they found is that approximately half of the later events were non-culprit lesions versus the culprit lesions. Again, supporting the same concept, the data shown below. So you've heard a little bit from Gabriel that there's some intriguing relationships between thrombosis and inflammation, and I want to build on that a little bit. And you'll be glad to know that we're not going to go through all of this, but there are several ways in which platelet biology is linked to inflammation. And I'm just going to illustrate some of the things in relation to CD40 ligand, to IL-1, to RANTES. And here's just an illustration of a cation-independent adhesion. This is from one of our PhD students who discovered a novel pathway of platelet biology during his PhD. So it's certainly possible to find some new things. And this just shows you that in diabetics, for example, there's upregulation of CD40 ligand and also platelet monocyte binding by another one of our PhD students. And then there was some recent studies that have looked at inhibition of thrombin receptors on platelets. And it's a novel concept because here's the N-terminal, here's the thrombin, here's the G-couple related PAR receptor, and it bends back on itself and amplifies itself. Unfortunately, I think those studies, like the TRACER study, got the doses wrong because there was too much bleeding. But there's no doubt that this does induce a number of changes like shape change, changes in inflammation within the cell. So we've got to think about the pathways in the cell and the pathways that we may be able to identify. So the challenge is, I've pointed out that we have multiple sites, multiple lesions. We can't identify these just in terms of the structural shape. Can we identify anything in terms of the behavior? And there are a whole series of studies that are underway to do this. As a crazy idea, we thought, well, could we identify some of these eroded or ruptured areas by localizing these disruptions using microspheres? Now, the trouble is you can't use commercial microspheres because they've got albumin in them, and so they bind all over the place. So we actually made our own with material that does not bind to anything, and then we put in a modified phospholipid shell and a linking molecule, and then a monoclonal. And the monoclonal is targeted, for example, at CD54 or TNF activated cells, so they don't bind to anything in an intact circulation, but only where you've got disrupted plaque. And in fact, it's possible with ultrasound to detect down to about a dozen of these microspheres. And we've done that in animal studies and in vitro. We've not done any of this in vivo yet. And we've got an even more crazy idea, which is delivery of the agents of interest from within the microsphere. Because if you can deliver it to the site at which you want activation, instead of flooding the whole system, you can then disrupt the microsphere and deliver it locally. So another crazy idea. Now, there's a guy called James Sharp in our university who was interested in developmental biology. And he's interested in the limb bud growth in the mouse fetus. You think, what on earth has that got to do with cardiology? Well, you know, that's the part of the excitement, is being able to develop integrated centers where we can think laterally. And anyway, he developed this technology by, instead of using conventional imaging, he used laser light. Laser light penetrates down to a cubic centimeter of tissue. And then he had the really smart idea of using CT algorithms to reconstruct these. And so that he could look at the way the limb buds grew. And what we did is say, okay, we want a different question. We're interested in blood vessels, and we want exquisite resolution of those blood vessels with laser light. Because it's laser light, it's many times better in terms of resolution than the current imaging techniques. So this is an intact mammary artery without it being destroyed, studied in vitro, and it shows you all the layers of this, and this is the intact fluorescence. And this is a transverse section, and you can see the endothelium, the media, right out to the adventitia as we track through this. Why is it interesting? Well, because we can put in fluorescent probes and switch them on and switch them off. We have to put in the probes outside of the green spectrum because some of the native fluorescence of fibroblasts are green, but we can do that. And one of my colleagues, who is John Mullins, who's interested in the some of the developmental biology in the zebrafish, has put in these reporter genes and used our technology to look at the way in developmental biology these light up and don't light up. And here's just an illustration of one of the intact internal mammary arteries, and the thrombus was within it. So we might have some tools to look at the way these develop. This just shows you a mouse heart with some pretty good resolution after ischemia reperfusion. Okay, so some tools. What about the genes? There's really a lot of challenges in polygenetic disorders that these are not influenced by single genes, but by multiple genes. We have huge variations in phenotype, and we've got gene-environment interactions. So we asked a question that if you were to draw this kind of schematic of susceptibility genes here about the disease initiation, and then disease-modifying genes later on. Environmental factors obviously influencing all of those. And then we wanted to concentrate on this end of the spectrum, because our patients are presenting here, not down there, at the age of 10 or 15. So we asked the question, within our large registry program, can we then ask a more specific research question by defining the genetic characteristics and observing the outcomes? And the rationale was that we know that we were able to determine some of the outcomes based on phenotypic characteristics with really fairly high degrees of accuracy, but weaker for MI. And that we may get some insights into these pathways of interest. Now, we chose, just as an illustration, this is not to try and tell you that it's only one particular polymorphism that's important. Absolutely not. But it's an illustration of how it can be done. There have been 4 independent genome-wide association studies that have shown the relationship of the 9p21 locus to the development of atheroma. What we asked was something different. Is there an association with the development of acute coronary syndrome, which we think there will be, and then in a population that's already enriched, are they even more susceptible to recurrent plaque rupture? And others have calculated that the population attributable risk, it may be as high as 20 to 30%. In the work done by Nilesh Samani and others, that's his estimate. So what we did is we took about 3,200 patients who presented with acute coronary syndrome and 3 distinct geographic populations. Because if we could replicate this independently in different populations, it makes it stronger, and a large number of controls, and that we had detailed phenotypic characterization and complete follow-up and looked at the outcome events. And the long and short is that both the heterozygotes and the homozygotes have a age, gender, and multivariable adjusted increased hazard of both the first event and recurrent events. So my friend said, okay, that's pretty interesting, but the trouble is it has to be wrong, because 9p21 lives in a gene desert. And 2 days ago, Bob Roberts said he doesn't think that can work because he doesn't know how it can amplify anything. Well, there was some interesting research that he hasn't read, and that's this. And that is that there's a very smart group from San Diego who looked at remote amplification. And this is published in Nature, and it shows long-range interaction with an enhancer locus. Now, is there anything of interest here? Yes, there is. Amplification of interferon gamma and STAT1. So there may be an elegant way that this and other genes may amplify some of the inflammation pathways modified by flow-related hemodynamic and other characteristics. Okay, I've said in the beginning we need to identify some vulnerable plaques. How are we going to do that? Well, we're fortunate in having a very well-developed imaging center in Edinburgh, and we've got various PhD students who are studying aspects of this. And this is from 320 slice CT, where you can define, for example, bits of calcium in the coronary arteries. And you can even look with altering the Hounsfield units and looking at plaques that are more likely to have lipid in them. But that doesn't distinguish it. And then one of our PhD students and then lecturers in our group, Mark Dweck, was interested in asking the question about atherogenesis and calcification in the aortic valve ring. And our group have previously done studies testing whether marked reductions in LDL would change the rate of progression of aortic stenosis. Unfortunately, it was pretty late to do that. And my colleagues have called this the ring of fire because it lights up on PET imaging. This is with 18-fluorodeoxyglucose, and we're able to match it with the excised aortic valves at the time of surgery. So that was quite interesting. And Mark won the American Society of Radiology Young Investigator Award for some of that work. And he was noticed as well that there's something very interesting, that some of the coronary arteries lit up. Now, this is very strange. The marker is sodium fluoride. Why should the coronary arteries light up? And we've gone on to do those studies, and Mark won the American College of Cardiology Young Investigator for the second batch of work. And this is published 3 weeks ago by Nick Joshi in our department in The Lancet. And this shows you the combined CT PET. So what I'm going to show you is combined CT PET with F18 sodium fluoride. Now, why don't we use fluorodeoxyglucose? Because there's too much in the blood pool. You've really got to look at carotids or peripheral vessels in order to differentiate it. So we decided that fluorodeoxyglucose was really not going to be the way to go. And we were interested in the concept that inflammation triggers microcalcification very early on. And this shows you 2 patients with acute myocardial infarction in A and C, obviously abrupt closures of this artery. And what you can see is lighting up of the lesion. These happen to be people who presented very late, but they presented with acute myocardial infarction. And then something quite interesting. We went on to look at patients with, for example, apparently stable coronary artery disease. And then if you're an angiographer, you look at this left picture and you see, okay, here's the angiogram. You can see this patient's had prior bypass surgery. They've got some irregularity, so there's obviously some disease in that, but no focal stenosis. And then you look at the CT, and the CT shows you that there's a fair bit of calcium proximally in this artery, and there's some calcium spread elsewhere. And then you look at the virtual histology IVUS, and the VH IVUS, for those not familiar with it, allows you to pick up fibrofatty tissue in green and some calcification in white, but the necrosis is showing up as the red area. And the VH IVUS in this yellow box corresponds, this is fibrofatty in this proximal area where there is some calcification, but in this area that looks completely inauspicious, you can actually see there's a plaque filled with necrotic material. Very interesting. So is this all hypothetical or is it real? So we went on to do a third arm of this study, which is to say we need real tissue to validate this. And these are carotid endarterectomy specimens. So we first of all did the sodium fluoride uptake, and then we've got the histology. And that links back to the approaches that Mike Davis took in validating his work with histology. And the histology shows at the top here, these are the F18 sodium fluoride positive plaques. That's the histology. Then interestingly, the alkaline phosphatase activity, which is upregulated, the apoptosis, and the cleaved caspases. Both markers of the inflammation that's disrupting plaque. Whereas if you look at other lesions that are negative, we didn't find this. So what's the significance of it? In our view, we really need to understand which are the plaques that are likely to progress. If we can understand that, we now have some novel approaches, including novel treatments that are aimed at modifying inflammation within plaques, and maybe profound lipid reductions to see whether we can switch off these pathways. And in my final slide, I would like to propose that we've made quite a lot of progress as a cardiology community in understanding ACS and atherogenesis. We understand something about the phenotype and something about the genotype and the pathways that are involved, and that we can characterize these phenotypes and pathways. But we now have some novel approaches really to identify some of the lesions that may progress in the future and may be vulnerable, and some interesting ideas about the role of microcalcification and inflammation in plaque, and new targets. So far from solving all this, we've got much more to do. Thank you very much.